So we are finally at the last video for chapter 4. At the beginning, I tried really hard to cut the length down, and I swear it is just not possible for this chapter. Anyway, I expect that this video should go a little bit more quickly because I feel like there's not a lot of explaining, because a lot of this is memorization. What do these other organelles, actually we haven't really talked about any of them yet, what do these organelles actually do in a eukaryotic cell? So bust out some flashcards, take some notes, and let's get started on these organelles. All right, so the biggest, most prominent organelle within a eukaryotic cell is the nucleus. The nucleus is really important because it is essentially the brain or the control center for our cells. It houses all of our genetic information. It is the location where our genetic information is taken out and turned into proteins and used to perform functions in the cell. So it's really, really important. Not only does it act as the control center, but it is also the storehouse. So it contains all of our DNA and therefore all of our genetic and hereditary information within this nucleus. Now, the DNA never actually leaves the nucleus, so the nucleus is really important in maintaining and replicating and fixing errors in our DNA. So there are 3 components to the nucleus. Let's actually start up here. This is a picture of our eukaryotic cell. You probably recognize this as the animal cell, but plant cells also have nuclei. So we've got a whole bunch of other organelles surrounding this. You might recognize the mitochondria. I mentioned the endomembrane system when we talked about invagination and endosymbiosis. All of these sort of pleated and squiggly organelles are part of the endomembrane system. There's a few other ones floating in there that we'll talk about soon. But this big purple one, purple for illustrative purposes, is the nucleus. Now, the nucleus is surrounded by, shockingly, a nuclear membrane. So that's this dark purple, this whole purple structure around the outside. It is a double membrane, just like the outside of our cell. And the very, very center of it is the nucleolus. Now, the nucleolus is basically the core of the nucleus, and that's where our ribosomes are produced. Now, we'll talk about ribosomes in a minute. I mentioned them already earlier. I think it was in our first video. Ribosomes produce proteins, so they're really important, and the ribosomes are produced here in the nucleolus. Then the rest of the area between the nucleolus and the outside membranes of the nucleus is our chromatin. So chromatin is just a fancy name for super condensed chromosomes. So the structure, our DNA molecules, are found in chromosomes. So a long piece of DNA is called a chromosome. These are thin fibers of DNA that carry all of our hereditary information. Now, we'll spend several chapters talking about DNA, how it works, what it is, what it does, etc. So we'll come back to that. Over here, we just have a picture of our phospholipid bilayer. You guys are familiar with that from earlier in the chapter. That makes up the nuclear membrane. And then we have pores, just like we did in our plasma membrane, that our messenger RNA, or mRNA, you guys might be familiar with that term. It's been in the news a lot lately. Our mRNA, which is little copies, short copies of our DNA, can actually exit the cell and be used to make proteins. So our DNA always stays in here, and only short messages in the form of messenger RNA actually leave the nucleus. All right, told you this is going to be quick. All right, we talked about cytoplasm a little bit earlier, and we said that that was the jelly-like fluid that surrounds all the organelles in the cell. It helps to give the cell its fullness and support. So it's sort of like the water in a water balloon. Without the water in the water balloon, you just basically have an empty balloon. But when you fill it with water, you give it structure and support. That's what our cytoplasm does. It also helps to suspend all of the organelles that are inside of it. So here's our cytoplasm in the kind of greenish blue color there, supporting and holding up all of those organelles within the cell. Now, within the cytoplasm, we have a cytoskeleton. So cyto meaning cell, skeleton meaning, well, skeleton. If you think about your skeleton, it is your bones, it is your support system that gives you shape, it gives you form. The cytoskeleton inside a cell does exactly the same thing. It provides a structure, a physical 3D structure for the cell. And without it, without the cytoplasm and without the cytoskeleton, that plasma membrane would just collapse on itself and none of the organelles could perform their functions. So you can sort of think of a cytoskeleton as like scaffolding inside of a cell. It provides support. Another function of the cytoskeleton is to help enable movement and also enables traffic within the cytoskeleton, or sorry, within the cell. So we've got shape and we've got movement. Now, you might think this is kind of weird that there's traffic inside of your cells, but your cells are doing things. They're doing all kinds of things, and we'll talk more about what they're doing later. But your cells are making proteins. They're making molecules, hormones, fats, things like that, products that need to be produced and shipped out to other parts of the cell. And so one of the ways that these products that your cell makes are moved to the edges of the cell to be exported is along tracts of cytoskeleton. Now, you also can have cytoskeleton that helps function with movement. So remember a little bit ago we talked about flagellum. We actually talked about flagellum with prokaryotes, but I mentioned sperm. So sperm have whip-like tails that move, that propel them in their environment. The cytoskeleton, these tubes that make up the cytoskeleton, can actually be connected to sort of machinery and attached to a flagellum, and that's basically a mechanical way for the cell to make its own movement. So cytoskeleton has two major functions. One is support, the other is movement. So over here on the right-hand side, I have some examples of the 3 different types of fibers that make up the cytoskeleton. You don't have to know the difference between them. I'm not going to ask you what each one does, but I just want you to know that they all help to provide support and movement within the cell. You do probably want to remember microtubules. They'll actually be important in a later chapter. And the microtubules are the largest fibers, and they do act as sort of tracks on which molecules can be moved within the cell. And in chapter 6, which is our replication chapter, we'll actually see how microtubules are involved in movement of chromosomes or DNA during cellular reproduction. So a little bit more on flagella and a related structure called cilia. So flagella and cilia are both long, thin projections that help with movement, but they have very different purposes. So cilia are short projections that beat swiftly. So think of this as like a rowing team all rowing together in the water. They beat together to move their boat forward. Well, cilia work the same way. They beat in synchronized motions to move fluids around the cell. So they're not moving the cell, but the cilia is actually moving fluids away. Earlier in the chapter, we talked about cystic fibrosis. In cystic fibrosis, the fluids on the outside of the cell are supposed to be moved by cilia. But because individuals with cystic fibrosis have so much mucus, that cilia actually cannot move all of that mucus and fluid away from the edges of the cells. So cilia move things around the outside of the cell. Flagella move cells. I've already talked about this a couple times, so I'm not going to say anything else except here are those sperm I've been talking about. All right, moving along to mitochondria. So we've already talked about mitochondria just a little bit. We talked about how mitochondria came about via by endosymbiosis. If you are not sure what that word means, you should go back and watch video 1 of this lecture. But basically, the endosymbiotic theory is the theory that states or explains the origin of the mitochondria and eukaryotic cells. So mitochondria were very early organelles, and they are the energy converters in our cells. So basically, they take sugar molecules and that have come into your body, been broken down and processed by your intestines, and the sugars taken to your various cells. The mitochondria take that sugar and convert it into usable energy for the cell, which is why we say that the mitochondria is the powerhouse of the cell, because they are the energy converters. That energy can then be used for a variety of cellular functions, including making proteins, making other products for the cell, movement, transport of those products within the cell. Pretty much anything that you could think of that requires energy is energy that is used, that has been being produced in the mitochondria. Remember that we said that support for the endosymbiont theory comes from the mitochondria having characteristics of a prokaryotic organism, including its own circular DNA, ribosomes, which are not really shown here, and then an outer membrane as well. And then, of course, that DNA is similar to prokaryotic DNA and more similar than it is to eukaryotic DNA. So mitochondria are very interesting in that cells that require more energy have more mitochondria. Now, this is true for several other organelles as well. When we talk about chloroplasts, the same holds true. Cells that are performing more photosynthesis have more chloroplasts, just like cells that require more energy have more mitochondria. So cells that are doing a lot of movement, like muscle cells, or cells that are doing a lot of processing, like liver cells, are going to require a lot of mitochondria because they have very high energy needs. Red blood cells actually have 0 mitochondria, so they are not converting anything, no sugars, into energy at all. So as a cell takes in food, there's going to be waste products that aren't used. There's going to be waste products that are produced from chemical reactions that occur in the cell, and organelles break down over time and need to be disposed of. This happens in an organelle called the lysosome. Now, the lysosome is basically a floating garbage disposal, and its sole purpose is to digest, recycle, and to get rid of waste products from the cell. So inside the lysosome are digestive enzymes and acid, just like your stomach. So it's essentially working like a stomach. It's going to break down any food particles, any used up cell components, and then transport the things that can be recycled to various locations where they can be used or moved to be dumped as waste outside of the cell. So here the lysosome is shown breaking up an old damaged mitochondria because they wear out after a short time. And it's just doing that by using digestive enzymes and acid. Just like our other organelles, it's surrounded by a membrane to protect everything else from its processes. So this is really important because if a lysosome bursts, it's going to release those digestive enzymes and acids outside of its cell. And you don't really want that. Over here, we have an organelle that's being broken down. This cell appears to have, this is the organelle. This is your digestive enzymes. This is an organelle that's being broken down within the cell. So your cells will also have multiple lysosomes. All right, so the bulk of the remaining membranes are part of the endomembrane system. And remember, we talked about very briefly that the endomembrane system came from invagination. Now that was a process by which the plasma membrane outside of the cell sort of pinched in and produced a system of membrane organelles. So that's the evolutionary history. The endomembrane system is a collection of organelles that are all related and all made up of essentially plasma membrane that's been modified over time. They're all part of a system, which is why we call them the endomembrane system. They are interconnected. They transfer products in between one another. So we've got the nucleus in the center here. Surrounding the nucleus is what we call the rough ER. ER stands for endoplasmic reticulum. So we have a rough endoplasmic reticulum and we have a smooth endoplasmic reticulum. If you look at that, you can see that the rough one has these little dots. Those little dots are ribosomes, which at this point, hopefully you remember, are the organelles that produce proteins. So ribosomes are stuck on the outside of the rough ER. So that should give us a clue as to the function of the rough ER. I'll let you think about that for a minute. We also have the smooth ER and the Golgi apparatus. So you might notice that these are all similar. They do look different, but they are generally similarly shaped. And products from the rough ER or the smooth ER are taken in little transport vesicles. Remember, those are like little pockets. They are taken and transported along the cytoskeleton from the smooth ER or the rough ER to the Golgi apparatus, where they fuse, and the Golgi apparatus does some processing of those molecules and then packages them up for transport elsewhere in the cell or to be excreted from the cell. So the smooth ER and the rough ER each make different products. We'll talk about those in a minute, but they make their products. They are taken to the Golgi. The Golgi modifies and completes those products and then ships them via vesicles and the cytoskeleton system to the edges of the cell. So there are two main functions of the endomembrane system in general, and that is the production of modification of molecules. So the endomembrane system produces things like proteins and lipids, and then modifies them and packages them up for distribution. The other function of the endomembrane system is to break down toxic chemicals and chemical byproducts, and then those can also be released from the cell as needed. So again, we have three parts. We have the rough ER, the smooth ER, and the Golgi apparatus. I know these are weird words, just bear with me. This is why memorizing comes in handy. So the rough endoplasmic reticulum. This is the one that we see surrounding the nucleus. Now, the reason it's surrounding the nucleus is that our DNA encodes information to make proteins. So our cells take information in the DNA, turn it into mRNA or messenger RNA. You don't really have to know that yet, but we're going to know it, so I'm going to say it here. Turns it into mRNA, and then that mRNA goes to the ribosomes, these little dots that stud the rough ER, and that mRNA at the side of the ribosomes is turned into proteins. So the function of the rough endoplasmic reticulum is to modify the proteins that are made on the ribosomes, because remember, ribosomes are what make it rough. They're these little dots on the outside. So ribosomes are protein making machines, and once the ribosomes make the proteins, they are sort of secreted into the inside where they are modified and can be shipped to other locations, the cell surface or outside of the cell. So the function of the rough ER is to modify the proteins that are made on the outside on the ribosomes. The smooth ER has a similar function but produces different molecules. So the function of the smooth ER is to synthesize lipids such as fatty acids, phospholipids, which make up our phospholipid bilayer, and steroids. So both parts of the endoplasmic reticulum work in synthesis or modification, but they are making different molecules. And here we are making the fatty molecules, the lipids. The other function of the smooth ER is that it detoxifies molecules such as alcohol, drugs, or metabolic waste. Now, I think that the smooth ER looks like coral sticking up. It's nice and smooth because it doesn't have any ribosomes. So the smooth ER therefore has nothing to do with proteins. Because of its role in detoxification, liver cells have a huge amount of smooth ER because the liver's primary function is detoxification. You might know that people who consume large amounts of alcohol have a higher risk for liver disease. And that's because their liver is constantly performing this detoxification. And that's all happening at the smooth ER. So when products are produced in the rough ER or the smooth ER, they are oftentimes taken to the Golgi apparatus, this organelle that looks like a bunch of folded membranes, for processing. So I like to think of the Golgi apparatus as like a warehouse processing center. So it gets all kinds of important products, finishes them up, packages them, and sends them on their way. So the Golgi apparatus processes and packages proteins, lipids, and other molecules for export outside of the cell. So over here we've got the same the same organelle from the top. These are all vesicles that have come off of the Golgi apparatus and are headed to the edges of the cell or to other parts of the cell with whatever little packages inside them. So you can kind of think of these vesicles as like delivery vehicles. They're going to travel from the Golgi apparatus with their newly packaged and processed proteins or lipids to wherever in the cell or outside of the cell they need to go. So again, this is the same slide I showed earlier. This is an overview of the endomembrane system with the rough ER, smooth ER, and Golgi apparatus. All right, so we have focused mainly on animal cells, but plant cells have a few unique parts that are not found in animal cells. Most of the organelles are found in both plant and animals, but there are a few that are unique to plant cells. So plant cells have a cell wall. Now, you might recall that some prokaryotes also have a cell wall. The cell walls are a different composition between plants and bacteria. We're going to focus here specifically on the plant cell wall. So plant cells are rigid, and this makes sense because plants don't have bones. We stand upright, our dog stands upright because we have that bone skeleton inside of us that is giving us support. Plants don't have that. They get their structure and their support from cell walls, which are essentially little boxes that surround all their cells. So down here we have a cell. You can't see it, but there is a plasma membrane right around the edge. Then around it is a cell wall. Inside is your cytoplasm, your nucleus, and then all your little organelles and whatnot. The cell wall surrounds the cell and provides a rigid support structure. It also provides a buffer to sort of maintain a water balance. Inside the cell, plant cells store water in vacuoles. Come back to that guy in just a minute. They store water in vacuoles. And if a cell were to bring in too much water, it would actually burst. So the plant cell wall, because of its strength and its structure, actually keeps plant cells from bringing in too much water. But they do need enough water to sort of stay rigid. The plant cell wall also provides protection to the outside elements. We have skin that provides some protection. The cell wall provides that sort of protection as well from insects or animals that might want to eat the plant parts. This slide also talks about plasmodesmata. I'm not actually going to test you on this. Plasmodesmata are basically junctions between the cell walls that allow materials and molecules to pass between and water between cells, things like that. You don't have to know plasmodesmata for this class. So vacuoles are found in plant cells as well. So this is our plant cell image. Just like our animal cell, it's got the nucleus, the endomembrane system, mitochondria. It also has chloroplasts and this vacuole. The vacuole here is basically storage. The cell can store nutrients, it can store waste products, it can be a storage area for poisonous materials that the plant takes up, or it can also be deterrents for insects or other animals that might be poisons to deter them from eating them. It can also contain pigments to attract birds or insects, and then helps to provide some physical support. So if it's holding water, that's the main function of the vacuole in a plant cell, is to actually provide some rigidity, hold the water for the plant cell. But the vacuoles do actually store a whole variety of different things. All right, so chloroplasts. Chloroplasts are probably the main thing that makes a plant special. These little green guys here. Chloroplasts are what give plants their green color. And chloroplasts perform photosynthesis. Now, photosynthesis is where the plant converts light energy into cellular energy. Because light energy, while there's a lot of energy in it, is completely unusable for most cells, but it actually can be converted into and stored in the bonds in sugar molecules so that the plant can actually use them. The reason that they are green is that chlorophyll, which is the main molecule within the chloroplast, actually reflects green light. So that's why we see plants as green, because they have a lot of chlorophyll. So any part on the plant that you see that's green, usually the leaves, but can be the stem as well, any part that's green is actually performing photosynthesis, or converting light energy into cellular energy. Now, this is important because this process of photosynthesis is where all of the energy on Earth comes from. We get our energy from the sun, but we get it because plants and some algae, some bacteria are performing photosynthesis. They are converting all of that unusable light energy into chemical cellular energy that other animals can eat, and that energy is passed up the food chain. Chloroplasts were produced by secondary endosymbiosis. That's where we had a prokaryote consume what would become the mitochondria, and then one of the lineages from that consumed a chloroplast as well. And so we get mitochondria and chloroplasts in our plant cell. So this chloroplast would have been an ancestral prokaryote that could do this, that could perform photosynthesis. They do have their own DNA. They also have their own ribosomes, and they have the double membrane on the outside, just like prokaryotes. So this, I think, is a really helpful image in our new version of the book. It's figure 439. I would suggest that you take note of this and go back to it. Use your flashcards use this information to make some flashcards. And it's just a really great summary of where these different organelles occur, plant or animal or both, and what their functions are. So I know this was a long lecture. I'm really sorry. I did my best, but was not good enough this time. Hopefully you are still awake and ready to continue on with your week.